We are talking about cell biology, and one of the important cell organelles are called ribosomes. Ribosomes are present both in prokaryotic cells and eukaryotic cells. You are already aware of prokaryotic, that means bacterial cells and other cells, eukaryotic cells. And if you will consider a cell, that this is a eukaryotic cell now. Since eukaryotic cell has other cell organelles also, in prokaryotic cells these are absent. Now two types of ribosomes have been seen. Ribosomes are present in the cytoplasm of the cell. These are called cytoribosomes. Cytoribosomes, those ribosomes which are present in the cytoplasm of cell. And if you will take, for example, a mitochondria is here. This is mitochondria. Or you will take chloroplast if it is a plant cell. Right, these mitochondria and ribosomes also contain these mitochondria and chloroplast also contain ribosomes. These are called organelle ribosomes. Organelle ribosomes. Now, difference is there between ribosomes in the cytoplasm and ribosomes in the cell organelles. Here we will see that prokaryotic ribosomes are called 70S and eukaryotic ribosomes are called 80S. I will explain you later 70S and 80S. Here cytoribosomes in prokaryotic cells there are only 70S ribosomes. In eukaryotic cells, two types of ribosomes are seen. If these are cytoribosomes, these are 70S. And if these are organelle ribosomes, these are, if these are cytoribosomes, these are 80S. And if these are ribosomes in the organelles, these are 70S. That means equivalent to prokaryotic ribosomes. Now, what is this S? Actually, ribosomes were first discovered by George Palade. Earlier he called them Palade particles, then he changed their name as ribosomes somehow in 1955. These can be seen under electron microscope because these are very, very small. Now, Sievertberg was a scientist. When we isolate the ribosomes, we use a process which is called centrifugation. The process is used to isolate the ribosomes or other cell organelles, which is called centrifugation. And here in centrifugation, a machine is used which is called centrifuge. Two types of centrifuges are available. These are machines. Just you have a mixer grinder in your house. Here it is a centrifuge. One is called a normal centrifuge and another is called ultracentrifuge. These are two machines, power-based machines, normal centrifuge and ultracentrifuge. And there is a motor here, which is usually called rotor. When electric current is given to it, it begins to rotate in the circular fashion. And here we can fit some test tubes here. And when it rotates, the test tubes also rotate. Now, normal and ultra, these are the two machines. Normal can rotate about 10,000 rotations per minute. It has RPM of 10,000 rotations per minute. Yeah, that means in 1 minute it can move 10,000 rotations if a normal power supply is given. But the ultracentrifuge can have the RPM of 50,000. That means it can rotate at least 50,000 rotations per minute. Other cell organisms, if it is mitochondria, nucleus, chloroplast, etc., these can be separated by normal centrifuge. But if we have to separate these ribosomes, we need ultracentrifuge. So this ultracentrifuge is used to separate it, and the process is called centrifugation. And while centrifugation is done, since it is time limited, what time it will take to separate, Swedberg used that time, and he gave a unit S, Swedberg's unit. Now, Swedberg's unit S, Swedberg's unit S, it is equal to 1 into 10 raised to power minus 13 seconds. This is the unit. 1s is equal to 1 into 10 raised to power minus 13 seconds. This is seconds. That means if it is 70s, it will take the time for separation in the ultracentrifuge 70 into 10 raised to power minus 30 seconds. If it is 80s, it will take the time 80 into 1 into 80 into 10, this 1 into 10 raised to power minus 13 seconds. 80s. 80 into 10 raised to power minus 13 seconds for separation. So this is actually the time. 70s and 80s is the time it will take to separate when these are placed in the ultracentrifuge. Now ribosomes are important. They are called protein factories. They help in the formation of proteins, and whole cell is dependent on them. It is controlled by DNA. It's a long process. DNA is involved, amino acids are involved, these RNAs are involved. All the three types of RNAs: messenger RNA, transfer RNA, ribosomal RNA, and these. Now these act as factories. If you see their structure, you will see that each of the ribosome has two parts. Actually, these remain isolated. One part is somewhat bigger. It is dome shaped, just like this. Another one is smaller, a cap shaped, just like this. This is smaller part. This is bigger part. Since these remain isolated in the cytoplasm, this is smaller part and this is bigger part. To avoid that stress on the cell, these remain separated. But when protein synthesis is needed, it will come closer, and magnesium ions are needed for their association. It is important. Cell should have enough quantity of magnesium so that these two can come together when there is formation of proteins, which is called translation. This is smaller subunit, this is larger subunit. If this is 70s, the smaller one is called 30s and larger one is called 50s. Same, S is equal to 1 into 10 raised to power minus 13 seconds. That is, smaller one will take time 30 into 1 into 10 raised to power minus 13 seconds for separation. Larger unit will take 50 into 1 into 10 raised to power minus 13 seconds. You will not have to add them. If you will add them, these will come 80. So you should not confuse here. It is actually time. This will take 30 into 1 into 10 raised to power minus 30 seconds for separation. This will take 50. And when we will combine them, these will take only 70 into 1 into 10 raised to power minus 30 seconds, because these will be somewhat heavier. And if it is 80s, then this is called 40s and this is called 60s. These two subunits. So the two subunits are important, and when there is protein synthesis is controlled by DNA or our genes. Genes form a RNA which is called mRNA or messenger RNA. Messenger RNA will bind. Actually, it is messenger RNA which will help them in binding, just like this. This is messenger RNA. And then there are messenger RNAs involved. It will have a coded message. Then transfer RNA is involved. It will transfer the amino acids one by one. They will bind together to form a chain. Ribosomal RNA is here, which will form a peptide bond between them. And then when the polypeptide is formed, when a chain of protein is formed, in the bigger unit there is a tunnel. Just like this chain will move through the tunnel. And here you know that almost most of the ribosomes, they stand on the endoplasmic reticulum, on the cisternae of endoplasmic reticulum, and then the chain will enter into endoplasmic reticulum. While it is processed, it is converted into useful product. So this was the structure and function of ribosome. The main function is they help in protein synthesis. Okay, thanks.